Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Now, let me introduce the next speaker, Christof Koch. So Christof is better known than I am, so he needs no introduction. But what is not terribly well known is that he was my first graduate student back in Tübingen, in the Max Planck Institute, and then one of the first postdocs at MIT. And Christof is now director or president of the Allen Institute in Seattle. Christof.
Thank you very much, Tommy. Wait, microphone. Yeah. Thank you very much. He's my doctor father, and the first thing I have to disagree with my doctor father is the deepest of all scientific problems. I'm not going to talk about. I talked about it last year here at NIPS. Is the nature of consciousness. How does consciousness arise out of 3 pounds of highly organized and excitable matter in our head? Which turns out to be orthogonal to the question of intelligence. So today I'm going to talk about intelligence. Being that I used to be a professor, I still have this professorial habit, so I'm going to give a little bit of intellectual background that partially overlaps with Tommy. I'll tell you a little bit about what we know about the neuroscience of intelligence. By intelligence here, I really mean high-level planning, making model building, etc. And then I'll tell you what we are doing about this problem at the Allen Institute for Brain Science. So the intellectual roots of both neuroscience and AI, they used to be heavily intermixed. They used to be, in fact, the same roots going back to, well, you can trace them all the way back to, of course, Leibniz and his universal calculus, subsequently Turing and the symbolic logic. At the point at which they were still sort of joined at the hip is really, I mean, there are many points in view. I like to typically start with McCulloch and Pitts, their universal calculus, where they showed that using some very simple automata, you could find a neural network for any logical predicate. This was back in 1943. And then together with memory, you could instantiate any Turing machine. And they showed in their paper, this is their figure 1, some of the simple computation that you could do. And I think historically it was very important because it showed for the first time how high-level logic of the sort that particular humans do might be instantiated in human brains using very simple neural-like element. And of course, these ideas later were taken up by John von Neumann in his book, The Brain and the Computer, and were really at the heart or at the root of the neural network. Then it was already mentioned by Tommy. Probably the most influential work was Hubel and Wiesel, for which they would later get the Nobel-prize at Harvard, where they studied in cat visual cortex. They found these cells that subsequently we also found in other animals, including in monkeys and other mammals, so-called simple cells that compute the orientation, and then complex cells.
Test 1, 2.
Yes, thank you. And then complex cells that compute the orientation over different locations. I actually found here, because many of you probably have never seen it, this is one of the original movies. Oh, okay. Well, there's this hash that you can hear. Let me see. Those are. Nope. So what happened in the 30s and 40s, people developed amplifiers. They became very small, and so finally the brain became eloquent, right? Until then, the brain was totally silent, but now you could put a piece of wire into the brain, amplify it, and actually listen to the way neurons talk. And of course, people discovered very quickly there was this universal idiom, the action potential in all neurons. Yeah. That's how the brain used to sound like. But now with electrodes, we can listen to it anyhow. And this is how they discovered, using bars of light of variable orientation and width, you can find the receptor field. So you had a cat was fixating straight ahead, so the eyes were fixed, and then you can map out the proportion in visual space where this neuron can be excited from, and particularly here it takes an oriented edge. And then the idea that you go from, because it turns out in the input to cortex, neurons are not oriented, they tend to be circular symmetric. And then within cortex, you have this hierarchy. You have simple cells that just compute orientation. You have complex cells that compute orientation over different locations. And then the idea is this hierarchy goes on. And then this was first formalized by Japanese engineer Fukushima in the neocognition, where you have a series of these networks, simple to complex networks, that end up with high level primitives at the recognition layer. And of course, at the same time, people like Charlie Gross discovered phase cells in the monkey. And then this is work that Gabriel Kreiman, who's one of the organizers, and I did together with the neurosurgeon Isaac Fried. We discovered these so-called Jennifer Aniston cells that are cells, this is now in human cortex, that are highly specific to individuals. Each cell is selectively to pictures in a very much invariant way, including to text and a computer saying the name of the individual. Here it happens to be Jennifer Aniston, of people that you're highly familiar with. In other words, things that you are overly exposed to again and again, your brain wires up specific neurons. And the idea was that this just very naturally emerges out of a very deep chain of feed-forward networks where you have simple and complex. And this is really at the heart of Tommy's H-max model that he also just presented, where you have this series of linear, non-linear transformation, and you cascade this many times over, where at the top end you have recognition. And I think in terms of just feed-forward networks, in other words, if I show you an image and within 120 milliseconds you recognize it, within 180 or 220 milliseconds you can begin to initiate the movement to release a button to signal the correct identity or category of that stimulus. So those sorts of networks explain that very rapid feed-forward sweep quite well. And of course, they also given rise, this was last year at NIPS here, Google and Net give rise to these now, 22, and I'm sure this NIPS, there are probably 28 layers or more layers of processing that all do spectacular well to recognize images. So you could argue that at least at the origin of history of AI and neuroscience, they were together, or computational neuroscience, they were joined at the hip. And so recently they've diverged, given the tremendous growth both in neuroscience as well as in deep machine learning. So what is it that we can say about intelligence in the brain? So here I really mean intelligence as measured by psychologists, i.e. by things like fluid or crystalline intelligence, by things like IQ. IQ is a very stable measurement. You can do it. It's very reliable. You can do it over many years. People have done it over 60 years, where they track the same individuals and do the same sort of test consistently year after, after many decades. So it's something very stable. It explains a large, there is a weak correlation in this. Whatever psychologists measure with IQ, there's a weak but a highly significant correlation between brain size. Now, so it's weak. It only explains 10 to 20% of the variance. All the evidence we have based on on lesion studies particular, and more recently on fMRI or other physiological evidence really implies that it's neocortex. So it's this neocortical sheet that's responsible for what we call intelligence, and also for things like perception, memory, reasoning, and planning and thinking. So when it comes to cortex, there are many things that we have no understanding of. So for instance, it's a well-known fact, brains come into, on average, there's a distribution between men and women. Men have typically 100 to 150 grams more brain. This translates, people have done these counts now a number of times, into 19 billion cortical neurons for women and 23 cortical neurons for men. We have no idea, there's no apparent intelligence difference between the genders. That raises the question, why? Now maybe we think about sex with those 4 million neurons all the time. It strikes me as quite plausible. And of course, we can think what implication that has for intelligence or the lack of thereof. Now, it turns out very embarrassing for Homo sapiens sapiens. People recently discovered, because of course we knew since many years, since essentially the discovery of large elephants and gorillas and whales, that other mammals have brains that are much bigger than us. But we always said, well, that's okay, they have bigger bodies and we can compensate for that by doing brain body index, etc. But we, human sapiens, we exceed, because we are the smartest, we have the biggest cortex of them all, and the baddest, supposedly. Well, it turns out that's not true, so they've now counted it in a pod of 10 pilot whales, which is a misnomer. They're actually dolphins, and they have 37 billion cortical neurons. Not neurons in the entire brain, but cortical neurons. we don't really know why they have twice as many neurons as we have, yet we are the dominant species. It's an unclear reason. The other thing that we don't really have any understanding of, the Homo Neanderthal, again, this is not controversial, this is quite well accepted, had a cranial capacity that's probably translated into brain tissue 150 to 200 grams more brain. So it's not clear why the proverbial caveman that died out at the same time that we made it into Europe, roughly 35,000 years ago, why they died out, but its smaller brain cousin, i.e. Homo sapiens sapiens, survived. We simply don't know. So we don't really have a good understanding of the link between natural intelligence, particularly this high-level intelligence, and the underlying neocortex. What we are beginning to understand really quite well are other aspects of intelligence that could be subsumed under the name of pattern recognition, pattern matching, i.e. Vision, intermediate, low-level and intermediate-level sensory processing, in particular vision, where most of the work has been going on. So all of that takes place in us. In other animals, there are quite different structures involved. But in us, much of that takes place in cortex. So cortex, the way to think about it is 2 plus epsilon dimensional computational tissue. I say 2 plus epsilon because across different mammals, it changed roughly from a couple of square millimeters to something like 2,000 square centimeters or 4,000 square centimeters. So it changed roughly by 10 to the 5 in area, 100,000. The width changed only by factor 3 to 5. So in a mouse, it's maybe 800 micrometer to 1 millimeter, and in us, it's between 2 and 4 millimeter. But the extent changed tremendously. So in us, it's a pizza. The surface volume, 1,200 square centimeter, it's a pizza, 14-inch pizza, 2 to 3 millimeter thick, and then, of course, heavily, folded up, and you have 2 of them, one in the left and one in the right. So that's why I think of it as the 2 plus epsilon dimensional computational tissue. It's relatively uniform. So, of course, since 150 years, neuroanatomists and physiologists have found differences across the cortical sheet. I mean, V1 is not M1, it's not S1, it's not prefrontal. But, if you take off your glasses and peer at it, the organization is remarkably similar across cortical surfaces with lots of specialization. And so the question that many people ask, in particular us at the Allen Institute, sort of what is the canonical columnar operation performed by cortex that makes it such a match that makes our intelligence, human intelligence or intelligence of other animals so robust and so flexible? Let me briefly tell you a little bit about Allen Institute for Brain Science, because we're very different creatures than universities or than industry. In fact, we're somewhere between biotech and industry. We're not for profit. We started in 2003, and all we do is basic science, basic research in the brain sciences. We're rapidly expanding. When I came, we were 160. We're now 310 or 320, and in a couple of years, we will be 500. Our culture, as I said, we have a heavy emphasis on research. Research is what we do, and we have teams of people headed typically by people who came from universities, who are faculty at universities. But then we also have biotech. We have products. Our products are large-scale data release that we release to the world at large, free. You can right now go to all of our data stores and download everything. There's no sign-up. There's no IP. There's no paywall. And we focus on a few large projects that can be done at scale. And so just like industry, we have program management, we have milestones, we have SMART goals, we have all that instrumentarium that industry has. And we initiated, when I came with Paul, we initiated a 10-year, billion dollar program for building cellular level observatories in both the 2 species that we focus on right now is mouse and human cortex. And that's our budget. And we are all funded by the curiosity of one individual, Paul Allen. He really wants to know how the brain works. He also wants to know, he's also started, of course, a number of years ago an Allen Institute for Artificial Intelligence, and he just started an Allen Institute for Cell Science. He's just interested in using his resources that are considerable to further sort of basic science. And he just gave us this building. Last month we moved in, a beautiful building, glass building where we can all be housed. At least we can expand up to 700 people. So that's in Seattle. So what we are both big science, in the sense that our experiments and our teams are big compared to academia, not of course compared to LHC or other physical experiments, but they're big by conventional neuroscience standard. We work in very tight teams, which makes it's a very different culture at university, and that's also the biggest challenge we have. And everything we do is open. So we release all our data typically many years before we put them out in our publications, in our Nature and other publication and all of that. We have 25 people in IT that do nothing but do sort of the preparation of the product and putting it online and making browsers and tools and coordinate system and answer the questions of that the user community out there has. So we study these two systems, the mouse and the human. They differ roughly by a factor of 1,000, very close to 1,000. Whether it's neuron counts, it's 14 million cortex in mouse versus 16 or 19 billion in human. Whether it's the area, whether it's the volume or mass, it's pretty much 1,000 to 2,000 times bigger. So these are some of the things that we do. In fact, this just came out last week, where we do in the human, we do gene expression in 9 different regions. So we take arrays, microarrays in many different regions, in many different brains. We process it using a sort of a standard procedure that we hold constant over many years, where we can look at the transcriptional architecture of the human brain. And what you find is really, this is a vignette, it's a living vignette, it's dynamic, that you can look at on our website. It shows, you pick 2 places of those 900, any 2 places in the brain, and you ask, how many genes are expressed differentially here above a threshold, which I think is log 2. How many genes are differentially expressed, and that number is plotted as a color. So blue means there are no or very few genes that are differentially expressed. And so you go, you go your different brain areas. So for example, if you focus on the lower right there, the blue area, that's all cerebellum. So if you look within the cerebellum, there are almost no genes that are differentially expressed above the threshold level. Now, it's interesting because the cerebellum, of course, 80% of your neurons are in your cerebellum. Most of your neurons, to be precise, 69 billion of your neurons are granule cells in your cerebellum. And they're really very homogeneous. Same thing if you look in the upper left, with the singular exception of primary visual cortex, which is quite different, to first order the transcriptional architecture. In other words, if you look at actually which genes are expressed in the various parts of the brain, it's relatively homogeneous across cortex. It's quite different if you go between cortex and hypothalamus or basal ganglia, and it's quite different within the hypothalamus or within the basal ganglia, but within cortex and cerebellum, these are both laminar structures, very similar. Now then, of course, there are differences in cortex. If you just look at cortex and you do something like PCA, you can very nicely see that 50% of the variance you can explain by distance. In other words, think about this sheet, this pizza, and now I look at any 2 places in the pizza and I compare the genes that are expressed here and the genes that are expressed here. And half of the difference, there are of course some difference between those, and half the difference is explained by the distance. The farther away you are, the more the differentially genes that are expressed. And it really brings home the physical nature of this thing. I mean, it's a physical process that we used to think with, and it really brings home the nature of that. So the bulk of our people right now are occupied in this project Mindscope, where we're trying to understand in as much detail as is possible today, using today's science, everything that goes on as the mouse sees, including its visual consciousness. And so we are looking at mice here. We have 1 million retinal ganglion cells. They have 45,000. They project to 23 targets, the retina, just like it does in us. One of them is the LGN. Here it's very small. It's only 18,000. That's much smaller than the human. And from there it goes on to primary visual cortex, just like in the human, that has roughly 1/3 of a million. In total, there are probably 2 to 2.5 million cortical neurons that respond specifically to vision in the cortex of a mouse. Now what we do 3 large projects. We generate in thousands of mice now detailed connectivity at the whole brain level, not only vision, but among all the areas, cortex and subcortical. We do a very detailed characterization of individual neurons, their shape, their electrical behavior, and the genes they expressed, including models. And we build cortical observatories where we observe and put it outside for the community, the activity at the cellular level throughout the entire brain of these mice. And all of these use the same, we all use visual cortex of young adult mice. We all map everything into high-resolution 3D coordinate systems, so we know every time we record from a cell, every time we image it, every time we put a microelectrode in, we record it in this atlas that has 10 micrometer pixels and 320 million voxels for the entire brain. And of course, we all use this common transgenic animals that we widely give available. We shipped 17,000 mice to the community. And all of this flows into centralized and standard database and is ultimately delivered to the user community. That's our latest release of our three-dimensional high-resolution atlas in the mouse. So for instance, here, what we're doing, this is the original paper. There are many more papers coming out, where we take in one mouse, we infect either in a wild type or in a transgenic animal. We use a viral technology to infect a particular population of cells, let's say the layer 5 cells in primary visual cortex. And we then use two-photon imaging and two-photon tomographic technique to reconstruct, to cut the brain into slices, 140 slices, and to scan in those slices with a pixel resolution of 1/3 of a micrometer, then to informational, do annotation, alignment, etc., and to make all of that data accessible throughout the entire brain. So now you know where the neurons in layer 5, the output neurons, the layer 5 pyramidal cells, where they project through at this mesoscopic population level. And you do that for however many population of neurons you can identify in whatever part of the brain you're interested in. So here you can see this is the first release of the data atlas. So there are roughly 500 you can go across. Each one of those rows is one injection in one particular part of the mouse. And then you can see the first half is the projection, the strength, the logarithmically compressed amplitude of the signal. So it tells you about the connection strength on the same side of the brain. And on the right side, you see it on the opposite side of the brain, and it's whole brain. So you can immediately see at high resolution the whole brain connectivity atlas. That's essential for our models as well as for the community. So this is one of the most frequently used atlases that we have. Then we're doing this Allen cell type database where we go in slices, both human and mouse, where we cut slices under standard condition. We do a standard electrophysiological protocol between 30 and 40 minutes. We record all of that at high resolution. We then inject biocytin so we can do the reconstruction of the dendrites and axons. We use a variety of models, both point models, a variety of GLIF models or GLM models, as well as very detailed biophysical models. At the same time, we look at the mRNA transcript doing single-cell RNA-seq, or now SMARTA, so again, in that particular cell, we can understand the RNA that's being transcribed. Ultimately, we want to do that for a few reasons. We'd like to get at how many different cell types there are, and also, of course, we like to use all that information to do very detailed modeling of that particular piece of the brain. So modeling, as I said, we use this family of different electrophysiological protocols. We use pulses, RAMs, noise, frozen noise, pink noise, pulses, etc., to do both biophysical, very detailed models, as well as that's of a larger interest to the larger computational neuroscience or physics community that wants to use the GLIFs to model networks. And then we have automatic procedures for fitting this data. So here you can see these biophysical, very detailed models using Neuron, where you can model both the dendrites as well as active and passive currents. The active currents we're doing in conjunction with the Blue Brain Project, Henry Markham, in the dendrites as well as in the soma. And all of this is available to everybody. Then here we have this paper coming out next month done by our transcriptional pipeline, our transcriptional group with Silke Tesig and Honghui Zheng, where we ask the question, so we know in chemistry over the particular work done in the late 19th century that every atom here on the planet falls into one of 92 discrete buckets, unless you're doing nuclear synthesis when you can shift things around. So the question is, in an adult mouse, or in an adult brain like you and my brain, is it true that every cell falls into one of a small number of buckets, and how many buckets are there? So we know early on people thought, well, there's excitation and there's a couple of inhibitory cell types. Well, now even if you look in the retina, there are sort of 75 to 80 different cell types. In Cortex, we found using purely transcriptional methods a smaller number, in this case 47, of which 42, a very nice number, 42 is the number of actual different cell types we have in primary visual cortex. Now it's interesting, this is half as many neurons as people have identified in retina. Of course, you could make the argument in the retina, where physics meets biology, you need lots of specialized neuronal structures to transcribe the 11 order of magnitude of light and map them onto output neurons that only has 2 orders of magnitude of gain. You need all these specialized neurons. But once the information is put inside the CPU, if you want, the central processor, you don't need that many different cell types. We'll see. This is early days. But so it's at least 42 different types of neurons, 23 inhibitory ones and 19 excitatory ones. And they not only have different genes expressed, but they have different electrical behavior. And in particular, they have different morphology and they project to different places. So the great moral of 20th century biology, and it's also true for 21st century biology, it's all about specificity. They're amazing specific gadgets. And the bad news is, if you want to look at pathological brains, you want to understand schizophrenia and Alzheimer and autism and ADHD, etc., all the other diseases we're prey to, those details all matter. Those details all matter because it appears that there's specific deficit, they affect specific types of cells. So it's not just good enough to know, well, it affects pyramidal cells. You have to know which type of pyramidal cells. And now all of this data is made available. You can go right now to brainmap.org. And you can download. So here, for example, you see the interface for the visual cell type database. This shows you the back of the mouse brain. You can click on any of the points. You can get a detailed list. You can zoom in and get a detailed list, for example, of the block phase imaging of the morphology of the FI curve. You can go onto the individual curves and access the individual electrophysiological recordings, and you can also download your models. So you can download either the point models or the biophysical detailed model. So it's not only, we're not doing this only to help us, because we have a large modeling department where we want to model all of that, but to also make all the data available to all of you. And we're also doing the same in human brains. In the human brain, you may know, every large neurosurgical practice, and we now work with 5 different groups of neurosurgeons, about once a month or so, a neurosurgeon will have to do an operation where he has to take out a part of a brain of a patient, because either there's a tumor there or there's an epileptic seizure focus. And typically what they do, depending on where the brain is, but in this case it's deep tissue. So let's say on this picture on the upper left, you see there's a tumor in the hippocampus, so they access the overlying temporal lobe and they cut that out. They sort of tunnel through, and the overlying temporal lobe is healthy, and then that piece of brain, which could be as big as the tip of my finger or my thumb, or sometimes it's like this, sometimes it's much less. That brain is typically, or a little bit goes to the pathologist. Most of it is discarded as medical waste. With the appropriate HIPAA compliance record and the appropriate technique, we can take that tissue and 20 minutes later have it in our lab. Now, 20 minutes ago, this part of the brain was part of a functioning brain and may contain its memory of its first kiss. And it's now there, right? So now what we can do, we can patch it. We can do physiology, we can recover the electrophysiological signature, we can keep these cells alive for 2 to 3 days, we can recover the morphology, we can reconstruct them, we can even use viral techniques and do things like channel rhodopsin. So in fact, we've infected these neurons with channel rhodopsin, for the first time shown, just like in a mouse or in a monkey, you can excite these cells using optogenetic techniques, human cells now. And then lastly, we are building, because we're interested in intelligence, i.e. in behavior, we're building these observatories. So here you can see part of the setup where we train mice under very routine conditions. There you can see one of our boxes where there's 6 mice that are all running on different setups. In our case, we do vision. So you have these little platforms like record table, and the mouse runs there and it sees different visual targets. And here we're training it for orientation selectivity. You could also train it for faces, to recognize faces. But here we train it to recognize different orientations, to stop and get water when it sees the appropriate orientation. So we call this brain observatory. And then what we do, so there you can actually see the mouse running. It's fixed in place, and we have a team of neurosurgeons that operates transparent windows into the skull, 5 millimeter windows, and then we can then put 2-photon calcium imaging microscopes over there. And these are transgenic animals where the electrical impulses, the action potential, are converted into flashes of light that we can pick up. And so we can literally peer at the mouse while on a single trial while it's thinking and making decisions to stop or not to stop. And we can routinely do this every time we can identify 12 visual areas. So we do that so we know exactly where we are. Now we can also do this new technique, which is really cool, which is single trial through skull imaging. There. So this is not average. This is through a skull. There's no transparent window in here. It's a GCamp. It's an AI93 mouse. This is what happens in real time without any averaging over the entire brain. As you can see the video of the mouse there. Below you can see its running speed. So it's and those little blue dots there at the 50 centimeters per second mark. That's when it gets reinforcement, when it licks and gets water. And that curve there is the speed. And so it's supposed to run past targets and stop at particular oriented targets. And here you can see 6 colorful curves, the response at that particular location, and in the middle you can see the overall brain response. And what you realize here, and this is never pointed out in all the existing literature on this, whether it's a mouse, a monkey, human, there's this brain-wide activity, and we have no freaking idea what it does, right? So typically what a paper will do, and there's now a string of papers every week in Nature or Science about, for example, visual cortex of mouse, which is down at the bottom there. It'll focus on activity in V1 and link that to orientation or to a reward or something. But then there's massive activity you can see in the front part of the brain, in the retrosplenial cortex, and we essentially don't know, and we don't know what it is, but you can see it on a single trial as a mouse does various things, whatever mouse do. As they whisker, of course, they move, their tail twitches, very complicated thing. So again, this data will all be made available. Finally, we're also doing very deep structural analysis where we're analyzing the tissue, because we really want to understand the circuitry. And the gold standard for knowing the circuitries, knowing the structures is at the level of 10 nanometers. So we're doing both light microscopy under Clay Reed, as well as array tomographic reconstruction under Stephen Smith, and collaboration with a large number of other people, including Andreas Tolias and Google and other people. They're just across from us in Seattle. And this, for example, is the array tomographic reconstruction, where you see the amazing depth of cortical tissue. This is a sliver, 80 nanometer thick, 100 micrometer across, and you can zoom in, and you can zoom in, and you can zoom in. The blue is sort of the Nissl staining of individual cell nucleus. So here we use antibodies. We can stain. Green are the excitatory cells, red are the inhibitory cells. So we come to rest at the level of synapses, and there are roughly 1 billion synapses per cubic millimeter. And so we'd like to understand the tissue. We functionally identified using observatories, and then we take that piece of tissue and cut it up and reconstruct it semi-automatically in order to understand the circuitry, and then link it up with high-level models. All right, and that is all right. And that was my second last slide. Yeah. So we hope over the next 10 years to contribute to a better understanding of. Let's skip that. All right. So with that, I show you our team. This is part of our team trying to understand how in both mouse and human, how intelligence and perception emerges out of cortical tissue. Thank you very much.
Many thanks, Christof. Since the room is pretty full, if you have questions, we have time for 1 or 2. Just raise your hand and I bring the mic.
Thanks, Christof. One question is about the chemicals in the brain, the stress chemicals, oxytocin, these things. I didn't hear any mention of, the effects of those, and any study, any data on that. Thanks. Very, very good stuff. Thank you so much.
Yeah, people
people, here we don't monitor stress hormones, so they would operate on the time scale of many seconds. And there's a variety of stress hormones. So you monitor during surgery, etc., the level of the stress hormone. Again, we don't know in detail what it does at the level of individual neurons. It does lead to an arousal effect that we can see. That's well understood. I mean, this is probably quite anxious for this little creature, you know, being fixed in the head, unable to move, and running past there. So that's why we do it, so it gets used to this situation.
Any plans in the future?
Well, I mean, so we do monitor under some condition these stress hormones. The trouble is there are many of them. There are many different hormones. There are many different arousal factors. There's dopamine, there's oxytocin, etc. And the question is, it's a very good one, how many of these factors do we need to keep track of? I mean, the bottom line is there are vastly more variables in the brain than we can possibly keep track of. So we have to select a small number that are representative and for the whole.
Thank you so much. That was fascinating. You showed an image right at the end of what looked like an electron microscope image with specific cells stained using a light microscope. Was that simultaneous imaging or something else?
Yeah, so we do both light and EM imaging using Apex, where we can actually use transgenic animals and do imaging of particular cell classes, and it shows up as an electron-heavy stain. So in other words, we can know in the EM, we can say, well, that's a GABAergic cell, or that's a cell that has a particular promoter. And so we use that. We use a combination of those together to give us additional information, what we call W-alpha beta, which is the cell type specific connectome. If there are 42 different types of neurons, there could potentially be 1,600, 42 squared, 1,600 different types of synapses, where cell type 27 has one type of synapse on cell type 28, but makes a very different synapse on a cell type 32. And we need to keep track of that to understand that.
And is that dense? So is every one
Apex is electron dense, yes, it is.
All right. Sorry, we're pretty tight in the schedule. We have a panel discussion later on, so any questions there? Or you can send us an email, brains.minds.machines@gmail.com.
Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.